Today I'm going to explain to you about the essential dogma of molecular biology. This is where we go from DNA to RNA to a full protein. And this whole process happens in two big steps called transcription and translation. Starting off with the DNA structure and the gene region. Here's my DNA. The top strand is called the coding strand and the bottom strand is called the template strand. Over here is called the promoter region right before TAC, and this is where transcription begins. ATG is the start codon and it signals where the gene starts being transcribed. GCA is an intron and it's basically a non-coding region that will be removed. TGA is the stop codon and it tells the cell where protein building instructions end. And after ACT, we have the termination sequence, which ends transcription. On to step 2, transcription. RNA polymerase bonds to the promoter site and moves along the template strand. Once RNA polymerase is done reading the template strand, it builds a complementary pre-mRNA strand. But the whole process isn't done just yet, because we need to remove the intron using a spliceosome. After splicing, we add a 5' cap and a poly A tail to protect the message. Here's the 5' cap. The 5' cap helps the ribosome find the message, and the poly A tail protects the mRNA from being broken down. Now, the mature mRNA leaves the nucleus and goes into the ribosome. This edited message now exits the nucleus and slides into the ribosome, where it will be read to build a protein. Step 3 is called translation, and this is the ribosome. It reads mRNA in 3-letter codes called codons. The first codon is AUG, and it is the start codon. It tells the ribosome where to begin. The first tRNA comes in with the anticodon, UAC, and it brings in methionine. This happens in the A site. It then moves to the P site, where the amino acid is joined to the chain. Here's the amino acid. Then the empty tRNA exits through the E site. Now more tRNA arrives, and the next codon is CCC. A tRNA with GGG arrives and brings proline. As you can see, and we place it onto the A site. Then it moves to the P site, and then exits through the E site. And then proline gets added to the amino acid chain. And then the next codon is AGU, tRNA with the anticodon UCA, and it brings serine. It goes to the A site, moves to the P site, and then exits through the E site. Serine gets added to the amino acid chain. Next is ACA. The tRNA comes in with UGU, goes to the A site, P site, exits to the E site. It also gets added to the chain. Then we have ACU and another tRNA with UGU, brings in another threonine, goes to the A site to the P site, to the E site, and exits. And another threonine gets added to the amino acid chain. UGA is the stop codon, and at the stop codon, a release factor enters the ribosome and tells it to stop building the protein, which ends translation. And now we have a full protein made from the instructions in our DNA. This is how our cells follow the instructions in DNA, transcribing them into RNA, then translating that RNA into a working protein. And that basically is the central dogma of molecular biology.